Proteins are polymers of amino acids. The general structure of an amino acid can be represented by an alpha carbon, which is the carbon that is neighbor to a carbonyl, that is attached to 20 different R groups or substituents, and is also attached to an amino group and a carboxylic acid group. So we're going to say that there are two different representations for the same general formula. We have the alpha carbon, which is this middle carbon, neighbor to the carbonyl, attached to an amino group in two different cases, but we want to indicate that this is a chiral compound because it has 4 different groups. 1 group, 2, 3, 4 groups, including a hydrogen. In all 20 amino acids, we will find that this part from the top will repeat. The only difference will be what is the R group. Nature is very selective and is using the L amino acids for protein synthesis. This is an L amino acid because the amino group is attached to the alpha carbon is to the left, opposite to what it was for the glyceraldehyde. From the 20 amino acids, only the amino acid of glycine is no chiral. That will be, we will have hydrogen twice, and if you have 2 groups of the same, that molecule is no chiral. From the L amino acids we said are used for protein synthesis during translation. That is part of the chapter of DNA and RNA. And the amino acids are found readily, but mostly in the cell walls of some bacteria. So it's not like they do not exist, but for protein synthesis, most of the amino acids that we will find are the L amino acids. And how is a polymer made? To make a polymer, we're going to have the reaction between the two ends of two amino acids. Let's pretend that this is amino acid number 1 with the carboxylic acid end free available for the reaction with the amino end of a second amino acid. So if we have amino acid 1 and 2, we can say that the reaction is similar to what we have seen in organic chemistry by the removal of one molecule of water. So we see we remove a molecule of water, this OH and hydrogen being removed as one molecule of water, making what is called an amide bond. But what is an amide bond in organic chemistry? In biochemistry, we're going to call that a peptide bond. And a peptide bond is represented by a carbonyl attached to a nitrogen. The hydrogen is always going to be trans to the carbonyl. And then the R parts that we see here, we have many copies of many amino acids attached together to make a polymer. That could be 100, 200, 300, but it's going to be called a polypeptide, and that will make a protein. Continuing with the discussion, before we can look at protein structure, we will discuss in a bit what is called primary structure, secondary structure, tertiary structure, and quaternary structure. I can just introduce and say that in the primary structure, that will have to do with what will be the sequence of the R substituents attached to the individual amino acids making the polymer. The secondary structure, that will be how the carbonyl and NH in the backbone are related to each other. And the tertiary structure is the folding of the protein. If we have the quaternary structure later on, we will see that that is when you have subunits that are also interacting with one another. But now let's take a look at the amino acids and their properties. The classification of amino acids, we're going to say the first thing is that's the general structure of an amino acid. We will have a carboxylic acid end. You have a carboxylic acid group, but we remember that the carboxylic acid group, we are not placing the hydrogen, we are placing only the carboxylate group because it is in physiological pH donating the proton to the solvent. Also, we will have an amino group because we have in an amino group a set of 2 electrons, and that is a base, will acquire a proton from the solvent, so that will be protonated. So we say carboxylic acid donates a proton to the solvent, amino takes a proton from the solvent, and that will create what we later will discuss as a sweeter ion or a dipole. Now, in terms of the classes of amino acids, this is the picture that we have in Georgia State all over the places, and I want to remind you that you are loved, you are unique, you are talented, and that's why I love being your lecturer, instructor, professor, how you want to call me. All right, So let's go back to the discussion. We're saying here that in the classification, we will have some amino acids that will be non-polar. For the non-polar amino acid, the first one that I'm discussing is glycine. I can say that it's actually unique, non-polar or polar, who cares? Because it's so small that we will say that it's only a hydrogen. So it's the uniqueness of glycine is that it's very small, so that it can fit in very small corners and places in a protein. The second amino acid will be replacing. If we replace that hydrogen and we're just gonna add a carbon. If we have a carbon, we remember that carbons always need to have an octet. So let's place here a methyl. Then this amino acid, if you guys see, all of the amino acids will contain the basic structure. That is an alpha carbon in all of them, a carboxylic acid group, an amino group. Alpha carbon with the hydrogen, carboxylic acid group, amino. So I have written a few so we can do this quickly. So for alanine is when the R group is a methyl. For valine is when the R group is an isopropyl. So let's call this one is an isopropyl, that is the R substituent. For methionine, we will have a sulfur. So instead of being only 3 carbons attached to each other, we will have 3 carbons, but in between, we will have also a sulfur. And that is a particular amino acid that is important for methylation in biological systems. Instead of doing an alkylation, adding an alkyl group, we will use methionine to donate this alkyl methyl group. Very important for alkylation of, let's say, nitrogen bases during replication and translation. So we will have modification of nitrogen bases. Continuing with the non-polar, we see that these are non-polar because they contain no hydrogen bond donor or acceptor on the side chain. So that will be the side chain that are non-polar. We see just high content of carbon, none of these with opportunity for hydrogen bonding. Some more that are hydrophobic, that will be a phenylalanine. Very easy to make phenylalanine. We have seen alanine here already. So if we bring one part of the alanine, that will be the first carbon. So you say, yes, let's bring this carbon and then place a phenyl ring. That will make phenylalanine. So let's put a phenyl ring so we remember how easy it is to memorize this amino acid. Overlook. You guys don't have an exam in the classroom to write it out for me for the quizzes, but just to remember how easy it is to make phenylalanine. You just add a phenyl ring, remove a hydrogen to alanine, you have a brand new one. Now for the amino acid of tryptophan, for tryptophan we will have in tryptophan the CH2. Now that is going to be attached to like a 5-member ring. So let's put here a 5-member ring with a nitrogen. That's a heterocyclic amine. And then attach to a aromatic benzene ring. This is actually called the indole. You have that this nitrogen has 2 electrons that are part of the aromatic system. And then we will have, since this is aromatic, we need to have electron delocalization. All of the carbons are sp2 carbons, so that is an aromatic compound, and the name for this amino acid is tryptophan. The part that we see this ring is what is called the indole ring. More amino acids that are hydrophobic. If you guys see just a lot of, you might say, but that's hydrophilic. No, because these lone pair of electrons are part of the aromatic system. This hydrogen is not donated. So it is a hydrophobic amino acid. These two are aromatic and also hydrophobic. All of these in this page are hydrophobic amino acids. And now let's do leucine. Leucine will have 4 carbons. So let's place leucine. And it has only 1 chiral carbon, that is the alpha carbon. Only the alpha carbon will be chiral. We can put a star to remember that there is a chiral carbon there. And then we have an isomer of leucine that is called isoleucine. The only difference is that this second one will have 2 chiral carbon. And for that matter, we're just going to say that the second carbon is chiral because it has attached 2 different groups. So we already have one group that is different. That will be the first group. including the alpha carbon and all of that part. So this one is chiral because it has a methyl, an ethyl, and the big part. So it's unique because it has two chiral carbons. And we remember that chirality in organic chemistry is of extreme importance. So we have that. So I put the name. I don't have room for no more in here. We have already the 4 amino acids that are hydrophobic plus 4. That is going to be 8 amino acids. We have already probably missing some, but you guys still have a list you can take a look at on the textbook. So we say glycine has the hydrogen, alanine has the methyl, valine has an isopropyl, methionine that includes sulfur. I will ask you the properties of amino acids on the questions. Leucine that has 4 carbons, and isoleucine that is an isomer but it has 2 chiral carbons. Now continuing with the discussion of amino acids, we look at polar ones. And among the polar ones we have serine. This polar is like if alanine had lost 1 hydrogen and we replace that with an OH, that will be serine. Now another one that contains OH and is also polar because we know that polar ones, they need to have hydrogen bond donor and hydrogen bond acceptor properties. And in this case, let's bring threonine. Threonine also has an OH and it also has 2 chiral carbon. Of course, the alpha carbon is chiral and the 1, 2, 3 carbon, third carbon from the carbonyl is also chiral. So we have 2 chiral carbons. So that is another part that makes it unique. Also has an OH. And the last one that has an OH, that will be tyrosine. Tyrosine is like if we already know phenylalanine. So we have a phenylalanine, and to that phenylalanine we can add an OH that will be para to the carbon that is in line for the alpha carbon. So we have in here tyrosine, Tyrosine, this is tyrosine. We have a phenylalanine that you added an OH on the para position. Now we also have, so we have serine, we have threonine, and now we have cysteine. It's also polar because it has a sulfur. Remember the difference that in cysteine is very unique because it's the amino acid that is going to be oxidized to make what we call a disulfide bond. We have discussed that, and that is coming back in protein structure. So the next one that we have is asparagine. Asparagine will have 2 carboxylic acid groups, but one of the carboxylic acid groups on the side chain, on the R group, instead of being a carboxylate group, that will have an amide, so it is an amide. Likewise, glutamine, oh, that's what it happens. I was supposed to do tyrosine here and glutamine here, but let's do glutamine over here. So glutamine is similar to asparagine. The only difference is that it's going to have 1 extra carbon. So we do a cross first, carboxylic acid group, a hydrogen, an amino, all right? And now we're just going to increase in carbon, 1 extra carbon, and still we have an amide bond. So let's put the carbonyl one way, and then NH2 over here, and that will be also the polar and uncharged amino acids. Now we're going to see the basic amino acids, and for basic we have 3 amino acids to look for. The first one is called the lysine, second one will be histidine, and this will be for arginine. I'm making sure my space is being used properly. All right, so for lysine, that will have 4 carbons, and it contains 1 nitrogen. And because we know that it is a pH 7.2, this one is going to acquire a plus charge. All right, so we still have 1, 2, 3, 4 carbons on the side chain, and the nitrogen will be protonated, and that will be a pH about 7.2-ish. All right, now we also have, that's for lysine. This is lysine, has a plus charge. And then histidine, this is an amino acid that at pH about 6.4, after pH 6, it's going to lose the proton. But if it is a lower pH than 6 diisoelectric point value of this amino acid, lysine will have lost the plus charge. And that one contains 2 nitrogens. It contains 2 nitrogen. One of them is going to be protonated. The other one is not, and it's also one that has aromaticity. So this nitrogen doesn't have a hydrogen. This one will have a hydrogen. You please take a look at those tables of amino acids, because I will ask you properties questions, properties of this amino acid. Now, arginine will have the same number of carbons than lysine, but it will have a center carbon that is attached to 3 nitrogen like this. So it's going to have 4 carbons. So we're going to place 1, 2, 3 carbons on a line, and then we have a nitrogen that is attached to 3 carbons. So we have 1, 2, 3 carbons. Okay, so that will be, I'm sorry, this is wrong. It's supposed to be a 1, 2, 3 nitrogen, carbon. This is a hydrogen, nitrogen, nitrogen. So we have one of those will acquire a plus charge. So we have, as a matter of fact, is NH2, one with a plus, and then this one has a hydrogen. So this one has a hydrogen. So that is the center that is going to have 3 nitrogens attached. Now, for acidic amino acid, this is arginine. For basic amino acid, this is arginine, histidine, and I didn't place the plus charge in there. And then we will have aspartate that has 2 carbons on the side chain, one with an extra carbonyl, and then glutamate that has 2 extra carbon. All right, so we have aspartate and glutamate. So for aspartate, it's very close to asparagine. This is an amide, and that's the second carboxylic acid. Glutamate is very similar to glutamine, the same number of carbons. The only difference is that one is a carboxylic acid, the other one is an amide.